Hey all, welcome back to Organic Chemistry 1 with Tara and Ray. As with all our videos, before I get into the experiment, I'd like to start with some general safety. Hopefully by now, you can tell Tara is not properly prepared for lab. She has loose hair, baggy clothes, and no lab goggles. The one thing she does have correct is closed-toe shoes and pants. Tara in this clip seems to be well prepared for lab, as she has her lab notebook, remember, don't bring your lab manual, a hair tie for her hair, as well as less baggy clothes. She also has a lab coat, which is optional, but very useful as it protects your clothes against any chemical spills. Notice she also has lab goggles, which are provided to you in lab. Once she's fully prepared, she's ready to go and start. Before starting the experiment, she checks her lab notebook to see what's needed. Before grabbing any glassware or chemicals, she first grabs gloves before touching anything else in the lab. Once her gloves are on, she's ready to start. As for fire safety, the front of the lab will always have a fire extinguisher and a fire blanket to cover any flames. It's very accessible. There, Tara's showing you the spill kit. A quick refresher on the safety shower and eye wash. In the back of the room, there's an eye wash that's readily accessible. Pulling it down activates it. And if you have spilled any chemicals on you, there's a safety shower. There's a curtain for your privacy as well. If you break any glassware, please let your TA know ASAP. They will help you clean up the situation. Here, a bottle has spilled. Notice, Tara is cleaning the area, making sure to sweep up in a wide range to ensure all glass is picked up. She then goes to the floor to get any glass that has been on the floor. Notice she does not directly kneel on the floor to prevent any glass from cutting into her clothes. Once she cleans up the mess, she will dispose of it. Here, Tara brings it to the glass waste disposal, where she will empty it out and then return the cleaning supplies. For today's lab, we'll be talking about alkyl halide reactivities in nucleophilic substitution reactions. And before we go into the lab, let's go over a quick introduction about the chemistry involved in today's reactions. So before we start, I want to talk a little about the alkyl halide structure and reaction language. Here we have an alkyl halide, and listed, it points out the alpha carbon. Note that the alpha carbon is bonded directly to the halide, while the beta carbons are the carbons next to the alpha carbon. This is important when explaining the reactions, as sometimes you may need to explain which hydrogens you're abstracting from, whether it's from the alpha or beta carbon position. It's important to note that alkyl halides can undergo two major types of reactions, substitution and/or elimination. We'll be focusing on substitution reactions today. The substitution reaction is called a nucleophilic substitution reaction because the electrophilic alkyl halide forms a new bond with the nucleophile, which substitutes for the halogen at the alpha carbon. Because carbon can only form 4 bonds, the halogen must leave and is called the leaving group. Alkyl halides are excellent electrophiles because halogens share a polar bond with carbon, are polarizable, and form relatively stable leaving groups as halide anions. In the example that I show on the screen, 2-bromopropane is converted into propan-2-ol in a substitution reaction, and we'll get more into that mechanism. So in many ways, the proton transfer process of a Bronsted-Lowry acid-base reaction can be thought of simply as a special kind of nucleophilic substitution reaction, where the electrophile is a hydrogen rather than a carbon. On the left is the Bronsted-Lowry acid-base reaction, and on the right is the nucleophilic substitution. You can see in both reactions we have very similar players. So we have an electron-rich species, which is the nucleophile and base, colored in red, and it interacts or reacts with an electron-poor species, the electrophile/proton, which I have colored in green. And our leaving group is driven off, and we can also consider the leaving group in the Bronsted-Lowry reaction our conjugate base, which I've colored in blue. So as you can see, these two reactions are very much similar to each other. And these similarities can help us understand the nucleophilic substitution reaction better. So, what is a nucleophile? You guys should know this. A nucleophile is a species that donates a pair of electrons to form a new covalent bond. And how do we tell we have a good nucleophile? We can measure nucleophilicity by comparing reaction rates. A faster reaction, the stronger the nucleophile. And finally, what are the four things that contribute to nucleophilicity? Here I've listed charge, electronegativity, the solvent the reaction's done in, and steric hindrance. I'll go into those bit by bit. So I started off by mentioning the charge. And one thing to note is that since a nucleophile is a species that's donating a pair of electrons, we can expect that its ability to donate electrons will increase as it becomes more electron rich, and then decrease as it becomes more electron poor. So, as electron density increases, so does nucleophicity. So, a nice rule to remember is that the conjugate base is always a better nucleophile. Here, in the example you can see in that first table, I have H2O, NH3, and H2S. And notice its conjugate base is much more electron negative, as shown by that charge there. And you can see that in the further molecules shown below. We have the phenol and then the acetone molecule. As you can see, the deprotonated forms of these are much more electronegative or electric rich, so they would make a better nucleophile. So, in the second table, I talk about electronegativity. So, assuming an atom has a pair of electrons to donate, the ability of a species to donate that pair should be inversely proportional to how tightly held it is. So the key factor of determining how tightly held an electron pair is bound is the familiar concept of electronegativity. So, let's simplify this. As electronegativity increases, nucleophilicity decreases. It's important to basically restrict this application to the trend of atoms in the same row of the periodic table. So in the image above, you notice I have carbon, nitrogen, oxygen, and fluoride. We were going to compare all those elements to each other. If we were then looking at silicon, phosphorus, sulfur, or chlorine, we could only look at those towards each other. So let's take a look at the carbon, nitrogen, oxygen, and fluoride. So as you know, fluoride is highly electronegative, so it's going to hold onto those electrons very tightly. So that means that our carbon molecule is going to have the best nucleophilicity. So it's going to be the best nucleophile because it's more likely to donate its pair of electrons. So, nucleophilicity is not a property that's inherent to a given species. It can be affected by the medium it's in, or in our case, we'll call this the solvent. So, I'm going to talk about two types of solvents specifically, a polar protic solvent and a polar aprotic solvent. I'm going to focus on this slide on the polar protic solvent. So, what makes a polar protic solvent given its name? So that solvent can participate in hydrogen bonding with a nucleophile, which basically creates a shell of solvent molecules around it. So by doing this, by creating the shell, the nucleophile is considerably less reactive because everywhere it goes, its lone pairs of electrons are actually participating with the electron-poor hydrogen atoms of the solvent. So, the ability of the nucleophiles to participate in hydrogen bonding decreases as we go down the periodic table. So, let's look at the halides. So, if we go from basically fluoride, chloride, bromide to iodide, we can see that as we go down the periodic table, our nucleophilicity will increase. Why? So, fluoride is going to be the strongest hydrogen bond acceptor, and iodide will be the weakest. Because, as you guys know, the ability of nucleophiles to participate in hydrogen bonding decreases as we go down the periodic table. So to simplify that reactivity a little bit more, is the lone pairs of the iodide ion will be more free than those of fluoride, which results in higher rates and, in turn, greater nucleophilicity. I've also included a few polar protic solvents that contain hydrogen bond donors. So there I have water, multiple alcohols, so we have ethanol and methanol, and then carboxylic acids. These are all solvents that can participate in hydrogen bond interactions due to their oxygens and hydrogens. So now I have polar aprotic solvents, and one thing to note is that the nucleophilicity increases going up the periodic table. So in our case, the weakest one will be iodide in this case, and the strongest will be fluoride. So why? Why does this happen? So one thing to note is that a polar aprotic solvent does not hydrogen bond to nucleophiles in a significant extent, meaning that nucleophiles have greater freedom in solution. So basically, under these conditions, the nucleophilicity correlates really well with basicity. And as we know, fluoride ion is one of the most unstable halide ions, so it will react the fastest with any electrophiles in the solution. So, a quick example of polar aprotic solvents. So this is polar solvents lacking hydrogen bond donors. Here you can see acetone. Here we do not have anything that really can participate in hydrogen bond interactions. Then we have DMF, which is also known as NN dimethylformamide. Here, once again, we have a molecule that very well can't really participate in hydrogen bond interactions. Then acetonitrile and dimethyl sulfoxide. So these are solvents that are polar aprotic, and these will change what is a good nucleophile in solution. So another thing I want to mention is, why don't we care about non-polar solvents? So we know that like dissolves like. So if we want a reaction to even take place, we need to use solvents that will actually dissolve our nucleophile. Because many nucleophiles are charged species, ions, they don't really dissolve in non-polar solvents. So we're not going to really have a reaction take place. So that's extremely important to take note of. Earlier, I had brought up mention of the structure with the alpha carbon, beta carbon, and that's going to come back into play now when I'm talking about bulkiness and steric hindrance. So essentially, when discussing nucleophilicity, we're often discussing reactions at carbon. So we have to take into account that orbitals at carbon that participate in the reactions are generally less accessible than protons are. And that effect of steric hindrance comes into play. So essentially what we're looking at is that the bulkier a given nucleophile is, the slower the rate of its reactions, and therefore the lower its nucleophilicity. So if we look at several types of deprotonated alcohols, which I have on the slide here, in the sequence methanol, ethanol, isopropanol, and then T-butanol. So the deprotonated form of methanol is methoxide, just for your reference, is actually the strongest nucleophile. And here we have deprotonated T-butanol, so T-butoxide, is actually the poorest nucleophile. So these effects are all important to take into account when looking at nucleophilicity, and then in turn when we're looking at these substitution nucleophilic reactions. Okay, so what is a leaving group? A leaving group is an atom that is displaced as a stable species, taking with it the bonding electrons. And a quick shortcut on telling on how we have a good leaving group is we can see we have a good leaving group when it can be recognized as the conjugate base of a strong acid. For example, water being the conjugate base of the strong acid hydronium ion. So, leaving groups are sometimes negatively charged, sometimes neutral, and sometimes positively charged. So, we can't really include a charge designation for our leaving group species. But referring to the comparison between acid-base chemistry and substitution reactions, the stability of the leaving group is evaluated the same way we evaluate the stability of conjugate bases. So, when comparing the reactivity of electrophiles that vary only in their leaving groups, the leaving group stability plays a dominant role. So the electrophile with the more stable leaving group will be favored. the lower the electron density of the leaving group, the more stable it is. Neutral leaving groups are favored over charged leaving groups. When comparing charged leaving groups, apply the concepts used to determine relative stability of conjugate bases, which I've listed for you. Identity of the atom holding the charge, delocalization of the charge through resonance, inductive effects, and orbital hybridization. So now with the basics covered, let's take a look at the Sn2 mechanism of one of the nucleophilic substitution reactions. So I'm going to look at Sn2 first. So when we start with a molecule with a chiral center, an example to the image on the left, S2-bromobutane, this class of reaction results in the inversion of stereochemistry. So note, our starting product is S2-bromobutane, but at the end we end up with R2-methylbutanenitrile. This is extremely important to keep note of, and this will be explained in the mechanism. Notice that we had a new bond form, so basically the carbon-carbon bond, and the bond that was broken was the carbon-bromide bond. Now, let's look at the figure to the right. The rate law. We know that rate is essential for telling how good a nucleophile is, but the rate law can also give us information about the mechanism. And for Sn2 reactions, it is second order overall. So if we look at our figure below, we see the same reaction that I presented on the left, but a rate diagram. So here we're looking at the increase of the rate as the substrate increases, as well as the increase of rate for the nucleophile increasing as well. With each of these increasing the rate, we can say that this is a second order equation. And that's going to come into play when we look at the mechanism. So, when looking at the Sn2 reaction, looking at any of these reactions, I didn't really mention the electrophile. And it becomes really important as the type of electrophile you have determines whether you're going through an Sn2 pathway or an Sn1 pathway. And it's really essential for understanding this lab. So one thing I want to note for the Sn2 reaction is that if we basically look at the substitution pattern of alkyl halides, there's a dramatic change in the rate of reaction when looking at whether we have a secondary, primary, methyl, or tertiary electrophile. So we can see based on the figure below that smaller alkyl halides like methyl bromide are fast, while more highly substituted tertiary alkyl bromide substrates, the reaction doesn't even proceed at all, with its rate being less than 0.001. This is important when you're analyzing your data in today's lab. So keep this in mind. Reaction rate increases as our electrophile becomes smaller for Sn2. And that is going to be explained when we take a look at the mechanism in the next slide. So let's put everything together to understand this mechanism. So the best explanation we have for what happens in this reaction is that it proceeds through what we refer to as a backside attack. So this makes sense to us because we have seen that there's an inversion of stereochemistry. So the nucleophile approaches the alkyl halide 180 degrees from the carbon-bromide bond, and as the nucleophile bond forms, the leaving group bond breaks. So the result of this is that there is going to be a transition state because of the partial bonds that are formed in this reaction. So there's two partial bonds. There's a partial carbon to the nucleophile bond, and then a partial carbon to the leaving group bond. And you can see that in the figure by the dashed lines. This is technically how you would draw the mechanism as well. Note the geometry as well. Instead of a tetrahedral, here we have a trigonal bipyramidal. So this is a 5-coordinate carbon, if only for a few moments too. So this backside attack makes sense because the nucleophile attacks the substrate from the back in a single step, resulting in inversion of configuration. Also, this explains the bimolecular rate law because it depends on the concentration of nucleophile as well as substrate. This also explains the inversion of stereochemistry and explains sensitivity to steric hindrance of the electrophile that I mentioned before. So bulky groups slow down the backside attack, while non-bulky groups will allow it to happen more. So this is why it's called the Sn2 mechanism. And all these factors are important to help you deciding what reaction is going on in today's lab. So the other reaction that can occur is called an Sn1 reaction. And let's talk a little bit about it. So if we start with an enantiomerically pure product, these reactions actually tend to result in a mixture of products where the stereochemistry is the same as the starting material, retention, or opposite, which we call inversion. So in other words, we have racemization occurring. So below you can see there in the reaction, we show here our alkyl halide that once we have that bromide leave and we install our alcohol group, that there's actually retention of stereochemistry, you can see by the methyl, or inversion. And this is actually a lot different than Sn2, which will always result in inversion of stereochemistry. So there must be a different mechanism going on. So let's look at everything we analyzed before and see if we can use that information to understand the mechanism. So let's take a figure to the right where we're going to observe the rate law. So this is experimentally figured out. So here we look at our reaction, and it's important to note that the rate is only dependent on the concentration of the substrate, but not concentration of the nucleophile. So the first graph shows that our substrate, as we increase that concentration, the rate will double as the concentration of alkyl halide is doubled. But the rate remains unchanged when the concentration of the nucleophile is doubled. So there's a 0th order reaction rate in the nucleophile. So that's where we put this information together and come up that Sn1 reactions must be 1st order rate overall. This is actually really interesting because Sn2 depends on the concentration of both. So why might this reaction only depend on the concentration of substrate? We'll get into that. So when we change the types of substrate, the alkyl halides that we use in these reactions, we actually find that tertiary substrates, for this instance, as shown in the figure below, T-butyl bromide, are considerably faster than secondary alkyl bromides, which are in turn faster than primary. So this is actually completely different from the case of Sn2, where primary was faster than the secondary and tertiary hardly reacted at all. So when we take a look at this mechanism, that's going to help us understand why this is so. So the best hypothesis we have for this reaction is a stepwise mechanism. In the first step, the leaving group will leave, forming a carbocation. In the second, a nucleophile attacks the carbocation, forming the product. So let's think about this with all the steps that I mentioned before. So first of all, the slow step should be formation of the unstable carbocation, which only depends on the substrate, not the nucleophile. So furthermore, since the stability of carbocations depends tremendously on substitution pattern, so the structure of the electrophile, tertiary carbocations are much more stable than secondary, which are in turn more stable than primary. This explains the dependence of the reaction rate on substitution pattern. So any factor which stabilizes the carbocation increases the rate at which the leaving group can leave. It also helps us understand the stereochemistry. Since the nucleophile is flat, attack could occur from either face, which means that we obtain a mixture of retention and inversion products. This is therefore called the Sn1 mechanism. So here we have substitution, nucleophilic attack, and then unimolecular, in contrast with Sn2, which is substitution, nucleophilic, and bimolecular. So now that we've gone over Sn1, Sn2, let's compare them to each other and finalize everything about them. So on the right side, I have a chart that lists basically the Sn1 and Sn2 reactions, comparing the rate law, the biggest barrier to the reaction speed, the alkyl halide, so our electrophile and its structure, the nucleophile, the solvent that we're using, and stereochemistry. And then on the left I have a fun image that's explaining Sn1 and Sn2 reactions, which I'll get back to. So the first thing I want to get to is the mechanism for Sn2 is concerted. So what does that mean? Sn2 reaction occurs in one step, and both the nucleophile and substrate are involved in the rate determining step. Therefore, the rate is dependent on both concentration of substrate and of the nucleophile. But Sn1 proceeds stepwise. So the leaving group first leaves, whereupon a carbocation forms that is then attacked by the nucleophile. The second thing I mentioned is the big barrier for Sn2 is steric hindrance, while the big barrier for Sn1 is carbocation stability. So this is the major thing that can prevent a reaction from occurring. So in Sn2, steric hindrance. Because Sn2 proceeds through a backside attack, the reaction will only proceed if the empty orbital is accessible. So more groups that are present around the vicinity of the leaving group, the slower the reaction will be. That's why the rate of reaction proceeds, primary is the fastest, secondary, and then tertiary being the slowest. Sn1, the big barrier, is carbocation stability. Since the first step of Sn1 reaction is the loss of a leaving group to give a carbocation, the rate of the reaction will be proportional to the stability of the carbocation. That's why carbocation stability increases with the increasing substitution of our electrophile, or specifically the carbon. So tertiary will be able to stabilize it the most, while secondary so-so and primary the least, as well as resonance effects. In Sn2, the rate of the reaction increases going from tertiary, secondary to primal alkyl halides. For Sn1, the trend is opposite. So let me explain that a little bit more. Since steric hindrance increases as we go from primary to secondary to tertiary, the rate of reaction proceeds from primary fastest, secondary to tertiary slowest. And then for Sn1, since carbocation stability increases as we go from primary to secondary to tertiary, the rate of reaction for Sn1 goes from primary, slowest, secondary, and then tertiary, fastest. So remember, Sn1 and Sn2 reactions only occur for alkyl halides and related compounds. If the leaving group is directly attached to an alkene or an alkyne, Sn1 or Sn2 will not occur, and that's important when you're looking at the substrates for today's experiments. Then, Sn2 tends to proceed with strong nucleophiles. Sn1 tends to proceed with weak nucleophiles. So, Sn2, as I mentioned, strong nucleophiles, and this generally means negatively charged nucleophiles such as methoxy, the nitrile, RS, N3, and OH-. But Sn1 tends to proceed with weak nucleophiles and generally neutral compounds such as solvents as methanol, water, ethanol, and so on. Now the effect of polar aprotic solvents and polar protic solvents on Sn2 and Sn1. So the Sn2 reaction is favored by polar aprotic solvents. These are solvents such as acetone, DMSO, acetonitrile, or DMF that are polar enough to dissolve the substrate and nucleophile, but do not participate in hydrogen bonding. Sn1 reaction tends to proceed in polar protic solvents such as water, alcohols, and carboxylic acids, which stabilize the resulting charged carbocation that results from the loss of the leaving group. These also tend to be nucleophiles for these reactions as well. And then finally, when a stereocenter is involved, basically Sn2 proceeds through a backside attack. If a stereocenter is present, the Sn2 reaction will give inversion of stereochemistry. By contrast, Sn1 leads to the formation, if it leads to the formation of a stereocenter, there will be a mixture of retention and inversion, since the nucleophile can attack from either face of the flat carbocation. So, back to that image about the cats. So, here you can see in one image, in the Sn2, the nucleophile, cat1, forms a bond to the substrate, aka the chair, and at the same time, the leaving group leaves. And then in the Sn1 reaction, the leaving group, cat number 2, leaves the substrate, aka the chair, and then the nucleophile forms a bond, aka sitting in the chair. So I hope this all helps you in understanding everything going on for today's lab. Now I'm going to go into safety before I get into the actual experiment. So, about safety. I hope you guys know what reactions we're going to be covering today, but if you don't, just a quick refresher, we're going to be using multiple alkyl halides and doing two reactions, one with sodium iodide and acetone, and then a separate one with silver nitrate and ethanol. And just a forewarning, we know that ethanol and acetone are extremely flammable. Actually, most of the chemicals we're working with today are extremely flammable, so it's important to keep them away from heat. And we will be working with a hot plate today, so we have to keep that in mind as well. Many of these are actually slight irritants as well as carcinogens, so everything is to be done inside of the hood today. And you're going to see that in the videos. So remember, in future when working with these stuff, be very cautious. Ensure you always have gloves, goggles, and are working in the hood. Okay? Now let's get started with the experiment. Here you can see Tara has gotten a rack with all of her test tubes, and she has labels where she is going to add to the top of the test tube. That is to ensure that if these test tubes need to be heated later, that the label will not be wet. Once she's done labeling them, she will move to the hood where she will then add all the other substituents to the tube. After labeling all of her tubes, Tara is getting water to set up the water bath. Ensure that you don't fill up the beaker to the full maximum height, as you don't want to risk water getting into your tubes. Here, she is setting up a rack to hold up the thermometer to ensure she can keep the heat at a reasonable rate, because if we heat it too much, we can risk our acetone evaporating in one of the reaction mixtures. It's essential to start this before doing the reactions, as we will need the temperature to be at the right temperature during the experiment. Once this is set up, you can move on to the next step. Tara is working in the hood. Here she's going to use a glass pipette and a graduated cylinder in order to measure out 2 mLs of a 1% ethanolic silver nitrate solution into each test tube. Once she does that, she will then add 4 drops using the Pasteur pipettes into each test tube. In order to save some time, I will cut to the addition of the alkyl halides to the reaction tubes and then show the reactions going forward. So here, Tara is going to be adding 4 drops of each alkyl halide into each tube, and then once finished, she will then cap the tubes and then give them a slight mix or shake. So there are multiple ways you can do this. You can shake it slightly side to side, or another way to mix them is to hold the capper on the top of the tube and then flick the bottom of the tube to appropriately mix your sample. As you can see, Tara is finishing adding, and you can see some of the reactants have already precipitated. This is before mixing. We'll show you a quick close-up of them right after mixing. So now they've been mixed, and you can see Tara's she's currently mixing them and holding the cap down and inverting them. The other way I mentioned was to tap the bottom of the tube as well. And as you can see, not even 5 minutes have passed, and about 4 or 5 of them have already precipitated fully. So you can use this information to discern what type of reaction kinetics are going on. So the rest of this portion of the video will be fast forwarded, and we're going to show you the waiting period. One thing I want to point out is that we've changed our waiting period to 10 minutes, and then we did our subsequent heating reaction. I want to point out that we've kept the labeling consistent to what is in the book, so you can actually write about the precipitate that is shown here in your lab reports, and this information will be useful. So keep note of this, and keep note of any color changes that you see, as well as the precipitate. Okay, and I'm going to fast forward the next part of the video. So now it's been 10 minutes, and we're separating what has been precipitated versus what we're going to put in the water bath. So as you can see, we have 3 reaction tubes, and notice that we've added a separate beaker inside of this beaker. This is to help keep our sample standing and to ensure that we don't have to basically use more racks to hold the test tubes up. So here you can monitor this portion of the video to see if any precipitation occurs. It's okay for you to fast forward to the next portion where we show if there's any changes at the end. So now we're removing our samples, and as you can see, there are slight precipitation after heating at the temperature desired. So here you can keep note of all the samples and check their precipitation. Usually in lab, you and your partner could each do each part of this experiment and then monitor it on your own and transfer results. But to make the video more effective, we're just showing you how to set up again for the other reaction. So here, Tara's labeling her tubes and getting ready to add the corresponding solution as well as nucleophile. Note that we set up the water bath in the last experiment and it's still going, so this will go straight to the hood and show the addition of the next nucleophile. Now that we're in the hood, Tara will repeat the procedure again, but now she will add 2 mL of a 15% sodium iodide in acetone solution into each test tube. Once again, I will fast forward this portion of the video as it's pretty straightforward. Note that she is using a graduated cylinder to do her measurements to ensure everything is accurate. Now with everything added, Tara is going to add the 4 drops of each alkyl halide to each reaction tube. You'll note while watching this portion that precipitation occurs immediately for some of these alkyl halides. The tubes are labeled 1 through 10, so you can approximate which tube is what. Keep note of any color changes. And for your convenience, I have fast forwarded the next portion of this video. Once everything is added, ensure to shake the tubes properly to ensure proper mixing. Keep note if any precipitation immediately forms after mixing, and note any color changes. As you can see, some tubes have shown precipitation already without any mixing. This is important to note, as this tells you information about the reaction rate and the kinetics of the reaction. Make note that we once again let this sit for 10 minutes instead of the 5 mentioned in the procedure. And at the end of this portion, we then separate the tubes versus what has been precipitated versus what we will be putting in the water bath. Please observe this portion. Feel free to fast forward to the next section if you'd like, or the end of this portion to see what has been precipitated. As opposed to the reaction in ethanolic silver nitrate, this one, many of the reactions did not precipitate in the 10-minute period, so we will move on to the water bath. Here we have put in the 7 tubes that did not show very little precipitation. Here they will sit for 5 minutes before we take them out and observe any changes in precipitation. Feel free to pause at the end of this portion to make note of any observations that you'd like to include in your lab report. To make things a little bit easier, we've separated everything. Anything that has heavy precipitation has been moved to the back, and anything with light precipitation has been moved to the front. Let's talk about cleanup. It's extremely essential in this lab as we've done about 20 different reactions. So, in the back, in the hood, you'll see a waste beaker. Please take your tubes and dump all solutions inside of them. The caps that you have can be put in the solid waste container. There may be a separate container to collect the corks afterwards. Make note that the tubes should be rinsed with acetone before put into the waste bin. For this lab, there is a special glass waste bin that can be found in the front of the lab. It is in the hood. Do not put the glass waste in the typical glass cleanup container that is found outside of the hood. Remember, the glass everything must be dumped from it, rinsed with acetone, and then put into the waste container. Here, Tara is just cleaning up all of the samples. Once she finished cleaning up the tubes, you can see her dispose of the glass test tubes. Make note, she also closes the container once finished. Here, the test tubes can go in this special waste container. Once you are done, make sure any other glassware is rinsed and put to dry.